Hello, JB Moneybags Incorporation. Today, we will be talking about the isolation and application of GFP gene from the G-Funk plant into a topical cream for treatment of melanoma and musculus species. Today, I'll be talking about this with my team, including Gabby Patton, Minerva Rivera, and myself, Blake Sanchez. Our research question is, when applied in a topical cream, does the GFP gene decrease the rate of proliferation in melanoma skin cancer cells in M. musculus species? Before I start this presentation, I'd like to go over why this topic is important to the world of science. To begin, cancer is the second deadliest disease in the United States, right behind heart disease. And more specifically, skin cancer is the most common type of cancer found in people around the globe. 1 in 5 Americans develop a form of skin cancer by the age of 70. An advancement in this field of skin cancer research would prove beneficial for millions around the globe and give new hope for those with diagnosis. Development of a topical cream to treat melanoma provides insight into further research on skin cancers. Our research plan is to determine whether the application of a topical cream containing this GFP gene has a large enough effect on the growth and proliferation of melanoma cells in M. musculus species. Once this GFP gene is isolated, it can be inserted into a Micamod aldera-based cream applied to the mice, and biopsies of these mouse skin cells will be taken over the course of 8 weeks to track the growth and proliferation of the cancerous cells. Now, to make this therapeutic cream, we first need to obtain multiple copies of the specific gene that we call GFP. Now, in order to do this, we will need to conduct a preliminary experiment, and the main goal of this experiment will be to successfully isolate and clone multiple copies of a gene called green fluorescent protein, short-term GFP, that is obtained from a rare plant, Green Glow Funkadelicus G-Funk, found in the Amazon rainforest. We will be doing this using molecular cloning methods. Once the gene is cloned and isolated, it can later be used to create a therapeutic topical cream with aims to treat melanoma skin cancer. Step 1 of the preliminary experiment will be to harvest DNA from the plant of interest, which is Green Glow Funkadelicus, short-term G-Funk. Leaf tissue from the G-Funk plants will be collected and placed inside a chilled mortar with an extraction buffer to be ground into a fine slurry. The fine slurry of the leaf tissue will then be transferred to a chilled centrifuge tube and later be centrifuged for 5 minutes. Doing this will separate the DNA from the other leaf matter. Step 2 will isolate the green fluorescent protein, GFP, from the extracted plant's DNA. Restriction enzymes are used in this step to isolate the DNA fragment of interest, which is the GFP gene. In this experiment, restriction enzymes AGE-I and EcoRI will be used to isolate and cut out the GFP gene from G-Funk's DNA sequence. This fragment of GFP gene is now ready to be inserted into a plasmid vector, but first this vector must be linearized via a process known as restriction digest. To conduct this process, the same restriction enzymes, AGE-I and EcoRI, will be used to so-called open up the plasmid vector and linearize the plasmid to ensure the preparation for ligation. Step 3 is to create the recombinant plasmid that will be composed of the plasmid vector along with the cutout section of the GFP gene. The two molecules, including the GFP gene and plasmid vector, contain overhanging bases that contain a high affinity for each other. This allows for hydrogen bonding to occur and bring the two molecules together. To complete this process, a ligase enzyme will come in and be used to seal the two components into a recombinant plasmid with a phosphoester bond. This will seal the backbone of the DNA structure and complete the recombinant plasmid. In this picture, we have P-Glow, which is the name of our recombinant plasmid. And you can see our first restriction enzyme, AGE-I, located after the base pair 1074, and our second restriction enzyme will be EcoRI, located after the base pair 2063. Next slide. Step 4 will be the transformation of the ligation product into competent E. coli cells. This is done by inserting the GFP-containing recombinant plasmids along with the competent E. coli cells so the recombinant plasmid can permeate through the bacteria's cell wall. However, this large GFP-containing recombinant plasmids do not easily permeate through the cell wall on their own. To help this process, calcium ions are used to coat the phospholipid bilayers of the E. coli cells with a positive charge to attract the negative charged DNA. This is done by placing the E. coli cells in a sodium chloride solution. The E. coli cells will also be exposed to a heat shock procedure, in turn disorienting the bacteria's cell wall so that the recombinant plasmids can enter the weakened host. Step 5 of the preliminary experiment will be to plate and grow the E. coli cells containing the recombinant plasmids on selective media. In the image below, we show how the 4 different types of agar plates that will be used to grow these E. coli cells, each of them containing different reagents, including ampicillin, which will be used as our selective marker in the experiment to help isolate E. coli that did not contain the recombinant plasmid. We then have arabinose that can be added to the Petri dishes to screen for the successful P-glow carrying bacteria clones. The presence of this arabinose activates the function of the PBAD promoter in the recombinant plasmid and thus is used as our screening marker. Our last reagent will be Luria broth, which is used to give bacteria a type of food source to be able to grow and replicate. Step 6 will be to confirm the presence of the GFP gene within a colony of E. coli cells. To confirm the presence of the GFP gene within the host E. coli cells, a method known as colony PCR will be used. To do this, the recombinant plasmid must be isolated from the E. coli cells via mini-prep, which uses an extraction buffer along with centrifugation to separate the plasmid from the rest. Next, the plasmid will be run in a PCR using primers that were developed by our team. After this step, you can analyze the length and base pairs of your plasmid DNA to determine the proper identity of it using gel electrophoresis. The DNA will also be sent out to a lab to be sequenced in order to obtain a higher certainty that the host cell did indeed contain the correct plasmid. Next slide. Step number 7 is the liquid culture of GFP gene. E. coli colonies and commencement into the liquid culture will be done to make large copies of this desired GFP gene. One colony will be chosen to be inoculated into a liquid culture in a 37°C water bath of Luria broth and antibiotics to fast-track division and replication of the desired recombinant clones. Once inoculation has occurred for 72 hours, plasmid DNA from the E. coli cells will be isolated via the same method of mini-prep as mentioned in the earlier steps. Lastly, step 8 will be the protein purification via column chromatography, which will be used to purify the protein from the rest of the DNA material. Specifically, we'll be using hydrophobic interaction chromatography, which will be used to separate the proteins based on the polarity. The green molecules in this image below show the hydrophobic R groups on the recombinant plasmid molecules that will bind to the nonpolar outer region of the membrane. Meanwhile, the molecules of the GFP gene, shown in purple and gray, will travel past and down the column and thus become isolated from the rest.
Following the isolation of the GFP gene, we will insert the gene into an imiquimod-based topical cream. We have selected to use the model organism musculus because its disease development capabilities are similar to humans. We've decided to use a cream because it is a non-invasive treatment, a preferred method of treatment compared to chemotherapy or other practices. Initially, a selection of 10 mice will be observed in order to detect important differences, but not waste resources early on. There will be 5 mice treated with cancer cells and 5 mice that are not. Biopsies will be conducted prior to selecting all of the mice to ensure they are cancer-free. With the cream prepared, we will inject 5 mice with melanoma skin cancer cells on their left lower back area. This will be our experimental group. Our control group is mice that are not injected with melanoma. 2 weeks following melanoma injection, all 10 mice will be treated with the cream on their left lower back area. This cream will be applied 5 times per week for 6 weeks to ensure adequate concentrations of the cream are taken up by the body. 14 days after initial application, the first biopsy will be taken of the mice that are induced with melanoma. Additional biopsies will be taken 28 days after initial application and 56 days after initial application. The last application of cream will occur 42 days after the initial application. Next. This is a flow chart depicting the harvesting, ligation, and transformation that occurs in the preliminary experiment, leading up to the development of the topical cream that is applied to the mice. Next. This is a list of ingredients in the cream that we will be applying to the mice. They are the ingredients that make up the imiquimod cream that we are using as the base. There will be 50 milligrams of GFP gene containing E. coli cells for every gram of 5% cream. Next. In order to track success of the cream, we will use the biomarker PMEL and microRNA 136. There's a specific binding region on PMEL that microRNA 136 binds to. PMEL proteins in the PMEL gene are recognized by the immunohistological marker HMB45 antibody and are targeted by microRNA 136. We selected this combination of markers because microRNA 136 targets PMEL via a WNT pathway that may treat cancers due to its effects on metastasis, proliferation, and apoptosis. If there's a decreased expression of microRNA 136 in the mouse, then melanoma cells will develop and proliferate. However, if there's an increase in expression of microRNA 136 in the mouse, then melanoma cells will not develop and proliferate. In this experiment, we expect the application of the GFP cream will increase the expression of microRNA 136, decreasing the melanoma cell proliferation. Success would look like decrease in abundance of melanoma cells, which is marked by an increasing level of microRNA 136.
And so our group predicted in our first outcome that melanoma-induced mice would show no melanoma growth within the skin tissue after GFP topical treatment. It was also predicted that non-melanoma-induced mice would present mild side effects due to the topical base cream. This is predicted because since the topical cream is working, it's still causing symptoms, but not as severe. And so in this bar graph, it highlights microRNA 136 expression levels in melanoma skin cancer. And so as you can see, the blue indicates that there are high levels of microRNA 136, suggesting that there are little to no levels of melanoma growth within the skin tissue. And in this figure, it is highlighting our second outcome, suggesting that GFPT does not work against melanoma-induced mice and resulting in melanoma growth within the skin tissue. It was also hypothesized that mice that were not injected with melanoma would show mild to severe symptoms from the topical base cream. And this is because since the cream is not actively working against melanoma growth, it's just sitting on top of the skin, and since it's not fighting against anything, it's harming those top layer cells, resulting in mild to severe side effects. And so in this bar graph, we see that there are low levels of microRNA 136, suggesting that there are high levels of melanoma growth within the skin tissue. And so you could say that they have an indirectly correlated relationship between microRNA and melanoma skin growth. And so with that, with every experimental design, there are some pitfalls. Our first pitfall would be the lack of a control group, or perhaps not enough control groups. So we tested against melanoma-induced mice and treated them with GFP topical, and also applied that same topical to non-melanoma-induced mice. So it could be ideal to have a group of just melanoma-induced mice. That way we have that data and can compare melanoma growth between melanoma-induced mice that receive the treatment, those that only received the treatment, and then, of course, those that only received a cancer injection. Another pitfall would be the skin or punch biopsies. This method is a bit invasive and uncomfortable to the patient. It could also result in nerve, vein, or artery damage. And so that could potentially be a problem because it could affect the healing process of the wounds and may lead to infection, since we are dealing with patients that have already weakened immune systems because of the cancer. And so it might be ideal to do blood samples after each application, or to just do a skin biopsy at the end of the treatment. But just not as consistent biopsies. That way they have healing time. Another pitfall would be the duration of the treatment. Perhaps extending the time between cancer injection and first application should be longer. That way the cancer has a bit time to progress, but of course we don't want to wait too long because that could result in very ill patients, and that's not what we want. And so another suggestion could be extending the duration of the treatment itself. So it was advised to apply the cream 5 times per week for 6 weeks based on imiquimod topical cream, but maybe extending the time of the treatment would yield in better results or even worse results. And lastly, the experimental design of the topical cream could also pose as a problem, since we are expecting side effects to occur. It could be difficult, or we could mistakenly diagnose a rash or a blister or some sort of side effect as a symptom or an effect from the cancer, or vice versa. We could mistake melanoma for just a simple rash or side effect from the cream. And so it could be a more suitable method to use a GFP gene injection instead of the topical cream. And so all in all, we feel confident with our proposal. We stand by our work, and we believe that GFP topical could be a suitable treatment for melanoma skin cancer in the future. And this disease development between M. musculus and humans stands as a good candidate for testing this GFP cream, since they do have similarities in disease development, and the observed effects of the cream on the mouse could provide insight on effects on humans. And so with that, we hope that JB Moneybags Incorporate will consider and approve our proposal to further our research in battling melanoma skin cancer. Thank you.